A graduate of Trinity College Dublin. Derek completed his PhD at King's College London, and then he did a postdoctoral research position at the University of Washington, Seattle, and University College Dublin, before holding academic positions at Maynooth University and subsequently at Trinity College Dublin. His research is aimed at finding out how the immune system protects against or causes disease in humans, and how it can be manipulated for the development of novel therapies. His group is particularly interested in populations of innate lymphocytes, so NK cells, NKT cells, and gamma delta T cells, and also mate cells, so mucosal-associated invariant T cells. These cells are early regulators of innate and adaptive immune response, as we heard earlier from Daniel Olive, and may be trained to prevent and treat infectious and immune-mediated diseases and cancers. So Derek, thank you very much indeed for joining us today and for being very patient. And we look forward to hearing your talk entitled CD1d-restricted T cells in patients with esophageal cancer. Thank you.
Okay, thank you, Jeremy. Can you hear me and see the screen? Great. Firstly, I'd like to thank you, Jeremy, for the invitation to share some of our results at this really excellent meeting. And I'd like to, first of all, introduce the person who has done just about all the work I'm going to present today. This is Ashanti Mello, who is a PhD student who's recently completed. So it is her data, and she was working on esophageal cancer patients. And the focus of her research, the focus of our research is innate T cells, as Jeremy has just mentioned, but I'm just going to talk about one type, and that's natural killer T cells. Natural killer T cells, or NKT cells, are T cells that recognize glycolipid antigens presented by CD1d, and they are particularly useful because they respond rapidly with an innate type of response. And the ligands for the T cell receptor is this molecule here, CD1d, and as you can see, it's very, very similar overall structure to MHC class 1 molecules. The main difference being the large hydrophobic groove that accommodates the long acyl chains of glycolipid molecules. And there are several glycolipid molecules that have been shown to bind to CD1d and to activate T cells. And these are just some of them. We've got them. These are glycolipids that are isolated from bacteria or parasitic organisms, and these all have been shown to bind to CD1d and activate a proportion of T cells. However, probably most NKT cells are more notoriously reactive against self-antigens. Here's a selection of some of the self-antigens that have been isolated. Some of them are, these are particularly produced in response to cellular stress, which might be infection, or, for example, toll-like receptor ligation. But I'd like to first start by talking about a subset of NKT cells known as type 1 or invariant NKT cells. They're called invariant because they have this invariant alpha chain of the T cell receptor. That's V alpha 24, V beta 11 in humans. And these cells recognize this xenogenic glycolipid α-galactosylceramide, or α-gal-CIR, as I'm going to call it. And this is just one subset of human NKT cells, and all other CD1-restricted T cells are type 2 NKT cells. Now, type 2 NKT cells are much more difficult to study because they don't have invariant T cell receptors for which we can get antibodies to detect them by flow cytometry. But the important thing about type 1 NKT cells is their role in tumour rejection. And this was first published in, I think, 1997 in these two papers in Science. Firstly, that showed that this α-gal-CIR can bind to CD1d and activate natural killer T cells. They have V alpha 14 chains in mice, V alpha 24 in humans. And it was also shown that if mice depleted of V alpha 14 NKT cells were not able to mediate tumour rejection. So these cells are very important in tumour rejection. And that led to a very large amount of work looking at tumour rejection in mice. And over the last 2 decades, there have been many, many studies with many murine models that have shown that alpha-galactosylceramide initiates tumour rejection. You just have to inject it into the mice. It activates invariant NKT cells, which then go to the tumours, and then they, in a downstream manner, activate other anti-tumour cells, such as natural killer cells and cytotoxic T cells. And we know this requires NKT cells because this does not occur in CD1 knockout mice or J-alpha-18 knockout mice. That would be the junctional sequence of this invariant alpha chain. Or in mice deficient in lipid transfer proteins are not able to mediate this tumour rejection. But tumour rejection can again be transferred by the adoptive transfer of invariant NKT cells to these mice. And look, mechanistically, these iNKT cells are able to directly kill tumour cells, but probably their most important function is the transactivation of other cells via rapid cytokine secretion. And in particular, as I mentioned, natural killer cells and cytotoxic T cells, which are then become the main effectors in tumour cell killing. And over a lot of work over the years has shown that NKT cells are right at the center of innate and adaptive immune responses. And they're able to make cognate interactions as shown by these antigen presentation molecules. They're able to interact with B cells, macrophages, neutrophils, myeloid-derived suppressor cells, as well as dendritic cells. And they're also able to interact with other cells such as NK cells and conventional T cells via cytokine-mediated crosstalk. And in this way, NKT cells have a very large ability to control many other cells of the immune system through both activating or suppressive interactions. But back to what we were mainly looking at was NKT cells in patients with esophageal cancer. And we looked both at adenocarcinoma as well as squamous cell carcinoma. And we found that using flow cytometry with antibody against the invariant T cell receptor, or by using a CD1 tetramer loaded with α-galactosylceramide, we're able to detect a small number of iNKT cells. This is about 100-fold lower than you would see in the blood of mice, but it is still there, usually less than 0.1% of all T cells in the blood. And we found that these cells were quite significantly depleted in patients with esophageal adenocarcinoma, esophageal squamous cell carcinoma, and in gastric adenocarcinoma. And this is a pattern that's seen in most cancers. There's a very striking depletion of iNKT cells. And when we looked a bit further, we found that these cells are depleted before treatment, and they remain depleted after treatment, both in the 3 types of cancer that I mentioned. On the top, we're just showing all the patients we studied before and after treatment, and below we have a small number of matched patients which we were able to get before and after treatment. And you can see that the numbers don't improve with treatment. And this is with treatment either by, well, usually a combination of chemotherapy, radiotherapy, and surgery. The depletion of NKT cells from the blood of most patients with most cancers, together with all the anti-tumour properties that have been seen in mice, have led to a lot of different human clinical trials using alpha-galactosylceramide. Alpha-galactosylceramide can be injected into the patients. However, this was found to be safe and well tolerated. However, it did not have any clinical efficacy against the tumour. So another strategy was to isolate NKT cells from the blood of the patients, expand them with antigen ex vivo, and adoptively transfer them to the patients. And a further type of strategy was to isolate dendritic cells, monocytes, differentiate them into different dendritic cells, then load them with alpha-galactosylceramide and adoptively transfer these loaded antigen presenting cells back into the patients. And these were all met with mixed success. And a more recent strategy is to take these latter two strategies, taking NKT cells and expanding them ex vivo, as well as generating dendritic cells and loading them with antigen and transferring them both back into the patient. And to date, none of these treatments have been licensed, but they have shown a lot of positive results, which makes it clear that it is worth pursuing these strategies until we can get them to work. And taking it a little bit further, NKT cells now are being engineered with chimeric antigen receptors. So we've got CAR NKT cells that maybe can combine the tumor targeting properties of CARs with the anti-tumor effector activities of NKT cells. So this is all work in process in many groups. We've been more concerned, how can we improve the efficacy of NKT cell-based immunotherapies? And what we've been doing is we can take NKT cells, we expand them up ex vivo. This is very easily done by magnetic bead sorting, followed by flow cytometric sorting, and then expansion with alpha-galactosylceramide, or we could just use PHA. And it is within a few weeks, from a very small number of NKT cells, we can get several million, tens of millions of pure iNKT cell preparations. And we are able to take these cells and co-culture them with tumor cells, either primary tumor cells or tumor cell lines. We've also tried using HeLa cells that would be transfected with CD1d, or we've also used autologous dendritic cells and B cells to look at the crosstalk. And we can load these with glycolipids and look at the effector functions of iNKT cells. And one of the very striking things we've noticed is that although iNKT cells are, we call them invariant because they have a fairly invariant T cell receptor, they're quite functionally heterogeneous. And just looking at these flow cytometry plots, we found that if you look at the CD4 T cells, they are predominantly producers of IL-4 when you stimulate them with alpha-galactosylceramide presented by CD1d. Some of them produce interferon gamma and IL-4, others mostly IL-4. If you take the CD8s, they're mostly interferon gamma producers, but some produce both interferon gamma and IL-4. And the double negative for CD4 and CD8 are somewhat intermediate between these. And this is important because when we looked at individual NKT cell subsets based on CD4 and CD8 expression, we find that the CD8s, as I mentioned, are more likely to release Th1 cytokines, the CD4s release Th2 cytokines, and the CD8s also have the most potent cytotoxic activity, suggesting that it is the CD8 T cells that we really want to use for therapeutic purposes for cancer. However, when we expand iNKT cells ex vivo using alpha-galactosylceramide, the CD4s selectively expand. So if you look here in unexpanded PBMCs, the majority of NKT cells are actually CD8 T cells, but after expansion, and this is 3 weeks of expansion with alpha-galactosylceramide, we can see the CD4s selectively expand. And these are the methods that have been used in the clinical trials. And as you can see, we are expanding the wrong NKT cells. And there is evidence in some studies that CD4 NKT cells can actually promote tumor growth and have the opposite of effect of what we want. Another way we have looked at to try and improve the anti-tumour responses was to modify the glycolipids. And here's alpha-galactosylceramide, as we've shown it as a mixed Th1, Th2 response when used to activate NKT cells. But there have been other ones. There's the OCH, it's a slightly truncated chains to produce a preferable Th2 cytokine response. This was published a good while ago and was found to be very good in ameliorating experimental autoimmune encephalomyelitis. And this alpha-C glycoside, which is identical to α-galactosylceramide, except with the anomeric oxygen here replaced with a carbon. This has got a 10,000-fold better anti-metastatic effect in mice due to its polarized Th1 cytokine production. However, this does not stimulate human iNKT cells. So we took an alternative approach and activated NKT cells with this glycolipid. It's a thioglycoside analog, and it is identical to α-galactosylceramide except with the anomeric oxygen replaced with a sulfur. And we found that this had a very similar effect to α-galactosylceramide. It wasn't really better, but it was comparable. And that led to further modifications by my chemical collaborator, Changming Zhu, who developed this one, XZ7. And this was able to preferentially induce Th1 responses from NKT cells without Th2 responses, and they were cytotoxicity responses as well as Th1 responses, but not the Th2 responses. And further analysis showed that this glycolipid preferentially activated CD8 T cells, which we know are more likely to induce anti-tumour responses. So this led to what other NKT cells are there present in the natural form? And for this, we used CD1d tetramers, which we purchased from ProImmune, and we loaded them with various glycolipids that we thought would be likely to bind to CD1d, and used them as flow cytometry reagents to detect NKT cells in patients with esophageal cancer. Here's the 4 glycolipids we've used in our preliminary studies. Sulfatide has been shown to bind to CD1d and activate some human T cells, as is cardiolipin can bind to and activate murine NKT cells. So these are the ones we started with, and we found that sulfatide indeed was able to detect type 2 NKT cells, which were significantly more frequent in esophageal adenocarcinoma patients compared to healthy donors. And this, has previously been published by another group that Vδ1 T cells, this is a subset of gamma delta T cells, a subset of these can recognize sulfatide. And we then looked at Vδ1 T cells in patients and controls and found that they are not significantly expanded in these patients. So looking a bit further, we looked for the presence of Vδ1 T cells, and we found that the sulfatide tetramer, loaded tetramer, did detect about almost 40% of the Vδ1 T cells in patients, compared to less than 10% or 20% in healthy donors. And another one, tetramyristoyl-cardiolipin loaded tetramers, also detected a large number of type 2 NKT cells in esophageal cancer patients compared with healthy donors. And so whereas type 1 NKT cells are depleted from these patients, these type 2 NKT cells are expanded. And there has been work done in mice that has shown that these type 2 NKT cells can promote tumor growth rather than being anti-tumor cells. So we decided to have a look at their tumor properties. And what we did, first of all, we were able to take these 2 glycolipids, we added them to transfectant HeLa cells that were expressing transfected CD1d molecules, and looked at the outcome of and use these to activate Vδ1 T cells. And we found that these cells could produce very little interferon gamma, but they produced IL-13, and in particular they produced TGF-beta, suggesting that these might be like the murine type 2 NKT cells that might be able to inhibit anti-tumour immunity. So in conclusion, we have shown that type 1 NKT cells are among the first cells to respond to tumours, and they are quite efficient at killing tumours and activating other cells to that will also have anti-tumour activity. However, type 2 NKT cells may suppress anti-tumour responses. So we are now very interested in looking at the actual complement of glycolipids that can be used to load onto CD1d that might skew NKT cell responses towards tumour or anti-tumour. And finally, I'd just like to acknowledge a lot of people that have contributed to this work. Most importantly, as I mentioned, Ashanti Mello, but there's been a lot of these people have been nearly all PhD students. And I'd also like to acknowledge my collaborators and the funders for this type of work over the years. And thank you very much for listening.
Thank you indeed, Derek. Thank you very much. Great presentation. I'd just like to open the floor to questions. If anybody has any questions, please raise your hand, or you can put the question in the chat as usual. While we're waiting for those to come in, just a couple of points. How did you identify some of those ligands? I hadn't come across the tetramyristoyl-cardiolipin before. And the second thing was just in relation to the CD4 NKT cells, and obviously another part of the story that I wasn't aware of. And I was just wondering if you could sort of share a little bit more in relation to that part of the story, which, yeah, just to know about.
Regarding why we chose those glycolipids, we chose sulfatide and cardiolipin because they have been previously shown to activate human or murine NKT cells in other studies. And when we were looking for them, these were the control glycolipids that we were suggested to get. So that's the only reason. Tetramyristoyl-cardiolipin is not known to be present in human systems at all. So it'll not be a natural ligand. So that really is the start of it. We're trying now other ligands that we know bind to CD1d, and we're still waiting to get these results. Regarding the CD4, CD8 expression by NKT cells, this is very different from in mice, which we have double negative and CD4 NKT cells, at least invariant NKT cells. And they have slightly different cytokine profiles. But in humans, there's also the CD8 NKT cells. And we just thought, well, since this is really the best way of distinguishing the NKT cells into different subsets, we just looked at them functionally, and other groups now have confirmed that the CD8 T cells have the best anti-tumour properties and the more polarized Th1 cytokine secretion.
That's interesting. Thank you. Quirin, question for you.
Thank you very much for the talk. That was very, very interesting. I was wondering, apart from changing the CD1d ligands, do you have other means to kind of skew the expansion by co-stimulation or by cytokine signals?
Not really. We always use IL-2 to grow NKT cells, but to expand, we are able to grow just with feeder cells, with feeder PBMCs, incubate, put into culture with sorted iNKT cells, and they grow very well. They're actually, it took us a long time to get it right because there's a lot of manipulation of the glycolipids, sonicating, vortexing, heating, etc. But now in our hands, it grows very well. And yes, we are able to skew the function of, and for example, we found in the presence of IL-15, we're able to get better expansion of CD8 iNKT cells. So definitely the cytokines are going to influence the types of NKT cells we get.
Thank you.
Perfect. Well, thank you very much indeed, Derek. I don't think there are any more questions. Oh, hang on. Yes, we do. Nick, do you have a question?
Yes. Hi. Yeah. Thanks for the great talk. Just wondering about the natural ligands for the iNKT cells. Is there anything known? I mean, the alpha-galactosylceramide is this strange ligand that's been found. Has there been any progress on the identification of any of those?
There have been ligands that have been identified. I showed you them in that slide. And that is, it is very few, considering these cells are so highly conserved between humans and mice, indicating their importance. There's been very few ligands. So there have been those autologous and some bacterial glycolipids that have been shown. And they have been shown to bind to CD1d and activate iNKT cells. But whether they actually are physiological ligands. There was one that caused great excitement, isoglycoptrihexosylceramide, and it was of great excitement the natural ligand for NKT cells, but it was later shown that it wasn't produced in sufficient quantities in vivo to activate cells, NKT cells. So there's a lot of doubt. Really, we don't know. And as a result, just about all the work on type 1 NKT cells has employed this xenogeneic glycolipid alpha-galactosylceramide.
Yeah. And is there anything known about the reason for the depletion of the iNKTs in cancer?
Not really. They are depleted just in about all cancer patients. Presumably, it is some way of overcoming the anti-tumour properties, so some adaptation of the tumour cells, but we really don't know.
Okay. Yeah, interesting. Thank you.
Thank you.
Thank you, Nick. Thank you, Derek.